Hi, hello everyone. Welcome to the NETRI webinar session regarding the compartmentalization of our chip platform, and we're going to talk today about MEA Pain. Okay, so thanks for waiting and thanks for connecting to this webinar number 5 regarding the compartmentalized MEA Pain on chip platform. Basically, we're going to talk today, and more specifically our expert, Benoît Maisonneuve, the product owner for Biopharma and Clinical Translation, supported by Hélène Gauthier, Dr. Hélène Gauthier, biopharma scientist also, and they're both, have a high level of expertise in both the bioengineering and neuroscience. So before to give the words to Benoît, I would like to present you the program. The program is basically based on two axes. The first one is, of course, the presentation of our electrophysiological impact of a chemotherapeutic agent on sensory neurons. And we're going to talk about that specifically using the electrophysiology as stated. And then we will have a question and answer session. So feel free to ask your question during the talk, and we will be happy to answer in live with both our experts. So quickly, let's just land a little regarding what we have already performed. Basically, the latest webinar, which was based on a multifluidic approach, a 3D microfluidic device called Duplex. And basically, we addressed this during the middle of May. During this session, you have, if you have participated or attended to this session, you have seen that we have presented results regarding brain organoid on chip, and also that we have shown some proof of concept of irrigated skin on this platform. So at any time you can go for a replay, and we will be happy to share also more information about this previous session. So now coming back to the heart of our technology at NETRI and why our technology is unique. Basically, you know, and you probably also have experienced, we hope with satisfaction, the high throughput and interoperable solution that we have regarding our 2D technology. And basically, this technology is based on the ability to mimicate the anatomy of the human body, so which means the compartmentalization of the cells that we can grow in our devices. The other thing that we can do with our device is the very easy to use device. That means that there is no equipment or pumps that are needed for our devices to run, basically for the culture of the cells and also after the culture of the cells. So it's a standalone device by itself. And lastly, we have made this device, this plate, compatible with lab equipment, as we are basically manufacturing a 96 well type of format for our microfluidic devices. So the idea behind our technology is to use the neurons as a sensor, and more particularly as a biodigital sensor. That means that we want to address with our technology a vast field of applications that could address different organs, as well as be specific in terms of readouts, and more specifically if we talk about phenotypic readouts. And obviously we want also to be able as much as possible to have characterization that are capable using non-destructive responses on the biology. So that's some of the things that you can benefit from our devices. Lately, which is a long-term project, we are working on the digital library, so the ability to compare reference compounds to the compounds that you want us to test or you would like to test in our devices. And so with this digital library, the ability to predict models and also gauge the efficacy and toxicity of these compounds that you are testing. So I would like just to make you a brief summary of the technology by itself before we move to more specifically on the CIPN. So as I stated, the magic of using net-free devices is basically to be able to compartmentalize the cells like we have, or let's say the organs, part of the organs in our body. So basically, what we have here is we have a chip, and chip is composed of 6 wells. Each chip is composed of 3 channels, and those 3 channels are critically isolated. That means that you can grow separately the cultures without affecting each other with mixing the media and the chemistry, generally speaking, or the biological materials. The idea behind our technology is to be able to link the neurons with the other part of the channels. So if we take here on the right part of the channel number 3, some sensory neurons here represented, and their ability to grow and to project from an axonal standpoint. And we will basically help the axons to pass from channel 3 to channel 2 using micro channels that are extremely small, and only the axons can go through. That's where is our unique technology. So from this little picture here, you can have the axon projecting from their cell body on the right side on channel 3, going through the channel 2 and then through the channel 1, where you have all the type of cells. It could be here keratinocyte, it could be gut cells, it could be anything, or neurons to neurons, you know. So basically, that's the idea. So which benefit can you take from is each chip, of course, represent an experimental condition, and as we stated, and then we will see later on, one condition means also that you probably can get also several results with the same chip, and basically potentially chronicity depending on how the compound can affect the biology. So having said that, just a brief mention about the 3D architecture that we have already presented during our last webinar, where we can mimicate the vascularization in our devices. So if you need more information on that, we can come back, but that's not necessarily part of this webinar. So basically, as we say, the projection of neurons into our device is something possible, and we have different architecture for that. We have the ability to do co-culture. We have the ability to do co-culture with a shift that way that one of the channel 3, is a bit more eccentric compared to the channel 2, that allows more synaptic studies here and the capability to see how the projection goes. We have also the ability with the same technology to do triculture using TriaLink devices, which could be very interesting in terms of, for example, an NG application, but not only. So again, somas in one compartment, the projection also is moving from one compartment to the others. And by the way, the ability for us to record the electrical activity of the neurons through the MEA technology. So as a summary of the format that we can propose, we have two kinds of format, which call half plate, called Edge, which is a 8 chips format. And then we have also the Pro version, which is basically 16 chips format. And important to state is that the technology, MEA technology, is compatible with an equipment from Axion Biosystem. This equipment is called Maestro. So you need to have this equipment to run all plates in your labs. Or alternatively, we can run as a service, and that's what we do. The plates and everything in all labs, including the MEA readouts. So I think it's time for me to give the word to and the speech to Benoit. Up to you, Benoit.
Thank you very much, Thierry, and once again, welcome everybody. So today we're going to talk a little bit about the work we performed on pain, more specifically on CIPN, so chemotherapy-induced polyneuropathy. We're going to talk a little bit about how we got there. So the experiment we started with, and how little by little actually we moved from conventional imaging readouts all the way to functional readouts. But before starting and getting into the heart of it, just in case some of you are not really familiar with peripheral neuropathies. So peripheral neuropathies are a side effect that is quite common during chemotherapy that can affect 60 to 80% of patients. And that often leads to dose reduction or even treatment interruption. As you can imagine, it can be quite detrimental for the patients because the survival rate can drop significantly during a treatment change. Moreover, even if you do not change treatments, actually 20 to 40% of the patients can develop chronic neuropathy. So that means that they will actually have pain even after the treatment is done. And that has a significant impact on their quality of life. So there are a lot of different types of neuropathy that are linked to chemotherapy. But if we really looked at how the neurons are affected by chemotherapy agents, there are 6 main categories with specific mode of actions. We're not going to cover all of them today. We're just going to focus on one platinum compound, which is oxaliplatin, and one taxane, which is paclitaxel. What is important to keep in mind is that there are a lot of different things that already start to appear actually to try to have an idea and try to anticipate the neuropathies that can be caused by chemotherapy agents. However, the current animal-based in vivo models, they often lack translationability, meaning that the mechanism, the pain mechanism can be fairly different. So you may not be able to actually detect pain in an animal, and you will see it quite commonly clinically. On top of that, the regular current in vitro assay that exist often lack any anatomical relevance, and mainly that's because of a lack of compartmentalization. So meaning that you cannot actually specifically target either the soma of the cells or the axonal projections. The fact also that most other solutions out there do not use MEA in order to get a functional readout, as you will see a bit later during the presentation, can be an issue. So quickly, the way we started, well, like everybody actually, we started from imaging. So we started to carry out some experiments on our devices and try to image what was happening to have an idea to see if we could actually see any physiological changes that would indicate potential neuropathy. So to do that, we used our dual-link platform. So like what you can see on the screen, as Thierry mentioned, one of the big advantages of this kind of devices is that you can compartmentalize. So in this case, what we did is we put iPS-derived sensory neuron cells in the channel 1, so only the somas was there, and we waited until the axonal projections reached the channel 3. Then, depending on the mechanism of action that was that is known and is quite largely reported in the literature, we decided to apply either oxaliplatin or paclitaxel. So for oxaliplatin, we apply in the channel 1, so we apply it on the soma of the cells, whereas for paclitaxel, we applied it directly in the channel 3, so on the axonal the axonal projections. So when we looked at it, quite unsurprisingly, what we saw was that in both cases, the more we were putting of either paclitaxel or oxaliplatin, the more degeneration we were able to see in the channel 3. So even when we put oxaliplatin in the channel 1 on the somas, we could see degeneration in the channel 3. So when we focus a little bit, if we focus a little bit on the channel 3 by itself, so what we did was quantify the surface covered by axons in the third channel, but also we quantify the number of blebs, so the little drops you can see that tend to appear along axons when they are degenerating. And unsurprisingly, what we could see in dose dependent manner was that the more agents we were putting in the experiments, the more generation we could see. There was a dying back effect from the axons. What is interesting as well is that you could definitely confirm what was in the literature, meaning that oxaliplatin mainly has effect from the cell soma, whereas the paclitaxel affects more the axonal projections. So of course, from there, considering that we could actually see with our with the cells that we use, so with the cell model, we could actually see signs of neuropathy, we then move toward our MEA platforms in order to try to quantify any functional change. So the idea is the same. The only difference really is that instead of using the classic dual-link without the MEA layer at the bottom, we use the dual-link MEA device. And then according to the experiment, we could add compounds in the channel 1 or in the channel 3. In order to try to explore a little bit more before starting really the work on CIPN, we use both human-derived sensory neuron from Axel, but also some rat DRG cells from QBM Cell Science, in order to make sure that we could see either increase of activity or decrease of activity according to the kind of stimuli we were using. As you can see on the top right corner, for example, when you use lidocaine on both type of cells, you can see a significant drop in mean firing rate. What you can see at the bottom here is if you use veratridine, which is a common compound that can be used in order to look at to induce the hyperactivity of the neurons. Well, what we can see is, so on the left, so you have 3 raster plots at the bottom. The one on the left side is before the addition of veratridine. The one in the middle is when you add it. So you can see actually an increase. When you look at the electrode view at the bottom, you can see a significant increase in activity. And then after a while, when you let it rest, it comes back to a different pattern. So it doesn't really come back to what it was initially, but it dies down a little bit. What is important as well to keep in mind is when you start to use cells, of course, there can be some variability that are induced by the cells by itself. So it's why we try to stick with a very strict quality control protocol where we actually look at the activity of every device compared to our historical data, and we make sure that we separate them in groups that make sense to make sure they're homogenized properly before moving on toward the experiments. So first, let's have a quick look at the hyperactivity that is induced by the paclitaxel cells. So as I mentioned before, we put cells in the channel 1, so the soma. The axon grows all the way to the channel 3, where we can apply paclitaxel. And what we could see, and what is interesting here, is at very low concentration, actually a concentration too low to actually see any physiological changes in the devices, we could actually already see some functional changes. So if you look at the top left corner of the screen, you can see the basal activity. So that's before we actually put any kind of compound. Just on the right of this graph, you can see the vehicle. So that's actually, we start by putting the vehicle just to make sure that what we're going to see afterwards is not just because there is something that is added on the cells. And at the bottom left corner, you can see the response to paclitaxel, and you can see that there is significantly more activity than for when you compare it to the basal activity. What is interesting, and that's something we're still actually exploring, is when you look at just the one in the middle, so paclitaxel at 5 nanomolar, you can see that the activity is lower than what we could see with 0.5 nanomolar. That's mainly due, we think, because we added it sequentially. So we actually first added the 0.5 nanomolar, and then we added the 5 nanomolar. So we think there may be a desensitization effect that is happening on the second exposure. So that's something we're still exploring. And on the bottom right corner, you can see the recording when we use KCL. So that's actually something we used quite often at the end of our experiments. The main idea is that KCL actually just make the neurons fire, and that's a good way for us to make sure that the neurons are still viable at the end of the experiment, and we didn't induce any significant death that could affect what we will be recording. So really the takeaway here is that using MEA as a functional readout, we could detect some acute effects on sensory neurons without inducing any cellular degeneration. So for oxaliplatin, for the sake of this presentation, we decided to show you something a little bit different, which is really the cold sensitivity. So we started to wonder, well, we can see that there is a difference in activity when we add the paclitaxel. Well, what happened actually? How does cells that have been exposed to oxaliplatin react to various kind of stimuli? So same idea than before. We use the dual-link shift MEA the dual-link MEA, once again, but this time we put the oxaliplatin in the channel 1 in order to expose the somas of the cells. What you can see here, so if we looked at different time points of the baseline, which is before we added anything, and then we looked at the cell 72 hours later. And we looked at different kind of, let's say, thermal challenges. So 32 degrees, so it's a decrease in temperature. The 37 is the regular temperature, and the 41 degree, which is an increase in temperature. What you can see from the top graph is, as expected, the activity of the neurons that we are recording, so the axonal weighted MFR to be precise, increased with temperature, which was what was expected. But then we wonder, well, there is an increase in temperature with the temperature, which makes sense, but do they react more or less if they have been exposed to oxaliplatin? And that's what you can see on the 3 graphs that are just below, where we actually normalized the MFR that you can see on the top graph with the baseline. And what you could see is, while at 41 degrees, we didn't really see any increase in terms of reaction, how much they react and how much they are hyperactive with the temperature challenge, there seems to be an increase at 32 degrees Celsius, so during the cold treatments. So this is very exploratory. That's really something we tried just because we were wondering if we could actually see it. And so more data is still needed, and we're still working on it to confirm the results, obviously. But this trend is very interesting, especially when you actually look at the vehicle, when you see that at all the temperature, you don't see significant differences in between the baseline and 72 hours. So there are a lot of, as I said, there is still a lot of work that is ongoing on that aspect, but there are other things as well, of course. So we have a few different perspectives about work that are going to come out of our labs in the next few months. So definitely stay tuned if you are interested, if you want to know more about what will be released. So if we had to choose 4 main perspectives, I guess, the first one would be to explore the capability to use actually chemotherapy agents and pain medication simultaneously to see if we could actually detect an effect of lowering the pain because of some drug. The second one would be to co-culture compartmentalized sensory neurons with dorsal horns in order to try to move closer to the full pain-relevant circuitry. So not just the sensory part, but all the way where we are going slowly, bit by little bit, back towards the central nervous system. Of course, as Thierry mentioned, something we are working on for the future is the digital signatures. And of course, that would be very interesting to start to define the digital signature that are specific to each of these altered states. And especially when we start to dig a little bit deeper into the various mode of actions, that we can start to see if we can have we can discriminate between the various type of painful stimulation. And of course, we want to explore other types of neuropathic pains. There are many that are important and that we definitely should be exploring, because there is a real lack of efficient medicine in order to manage it in the long run in a lot of different pathologies. I think that ends my part of the talk. So thank you very much for your attention. And I think, and actually, as Thierry just said at the beginning, do not hesitate to send us some questions. We'll be more than happy to answer them at the best of our capability.
Thanks, Benoit. I would like to introduce you, Hélène, who is joining the group now. Thanks, Hélène, for coming. Very nice of you. And then we have basically a few questions that came up as Benoit was talking to. So there is a first question. Maybe, Hélène, you can help on that one about the compartmentalization. And the question was, how do you make sure that only axons, not dendrites, pass through the microchannels?
So there is 2 aspects, length, and this has been shown in a publication previously that axon cannot grow more than, I think, 400 micrometers in the microchannels. So you have the dual link shift that has been specially made to ensure it's only axons that will cross on the other channel. And also sensory neurons have really long axon projection and 2 main axons.
Okay, great. Thanks. We have another question that is coming from, still the platform and more specifically about the use of a platform. And can it be used to assess the risk of neuropathy from antibody drug conjugate? Maybe, Hélène, you can answer this one.
Yes, so our platform has been built, so first for the readouts that shows the dying back of the axons on the neurofluidic line. So any toxic neuropathy that has the same phenotype of, we will be able to replicate. So yes, any toxic neuropathy can be modeled on the platform.
Okay, very good. In terms of regulatory challenges, again, for talking about our platform, the question is, are there any regulatory challenges associated with the adoption of organ-on-chip technology? So maybe, Benoit, you could be the one to answer that.
Sure. So that's actually a really good question. So when it comes to efficacy, there's no real regulatory challenge. You can use the devices and you can use whatever results you have and integrate it into your application, really. It gets a little bit more tricky when you go towards safety, and there are still some challenges. I guess the more important one, really, they go around the standardization of the protocol and the methodology. Make sure we all have what we can accepted readouts that we all agree on, and reference compounds that make sense. So there are some challenges, but more and more the various agencies are getting very interested actually in the capabilities of these kind of platforms, and there's a lot of ongoing work that is happening actually in Europe, but also in the US, a bit everywhere on the globe actually, in order to increase the confidence and to make sure they get more and more used to the kind of data they can have, so they can get actually, at least, even if they won't be removing the need for animal testing in the first stage, at least can be used in conjunction to show that a drug potentially is safer than another one, for example, that another one that is on the market, for example.
Thanks, Benoit, for this extensive answer. Very, very lightening the for the regulatory challenges as we have today. So we have another question. So I will translate it in English. Have you tested the response of capsaicin on sensory neurons using Netri chips? So maybe I can give you both.
Yeah, please, Hélène, you go.
Hélène, maybe first.
Yes. We did. So we cannot show you the data because some of it has been done with clients, so we cannot show you. But in fact, the protocol that has been used to test the acute effect of the paclitaxel was developed to test the response to capsaicin. And yes, we do. And also, it's one of the criteria when we select suppliers, cell suppliers, that their sensory neuron should respond to capsaicin.
Just to complete on that, because I actually just mentioned about cell suppliers. So I mentioned the quality control we do in every experiment, but we also have quite an extensive quality control that is happening with the suppliers to make sure that the cells we receive are of quality and that they work as they should be working. Because when you think about organ on chip, well, they're all based on the cellular activity that's going to put in there. So that's really the heart of the model. So it's very important to make sure things work properly.
Excellent. Thanks very much for that. So maybe staying in the same subject around that one, but it's still the same subject, of course. But just one question. It's regarding the oxaliplatin versus paclitaxel. So the question is oxaliplatin inducing acute hyperactivity like paclitaxel? So Hélène, that's for you, I guess.
Yeah, that's a very good question, and I really would like to know. So that's it's in fact we were trying to develop the platform and test different modalities. So Can we test an acute effect? Can we test thermal sensitivity? So this is just the first step, but yeah, we would like to test also the oxaliplatin in the same way that we did with the paclitaxel in order to compare both compounds.
Okay, good. Another question I do have about our method is, would other types of neuropathy be possible to study with this method? Who wants to answer? Benoit?
Whichever. Yeah, sure. Yes, I mean, definitely that's kind of what we are planning. And it comes back to what you were saying before with the antibody drug conjugate. The idea really is we have a simple platform in a certain way that can already enable us to explore various modalities and various conditions. So we can imagine, for example, exploring, and that's something we actually are on. That is ongoing work. We are exploring inflammatory pain, for example. Later on, we would like to explore diabetes-related pains, but there are also other conditions, actually other syndromes that induce neuropathies that we all want also to explore.
Okay, thanks very much, Benoit, for that. We have more and more questions, so we're happy that it's going live, so we don't hesitate. We have another question, so the questions are not in the order, they come as they come, okay? So we have a question, have you tested TRPV1 and TRPA1 agonists? So can someone of you maybe clarify that?
In terms of TRPV channels, we only tested the capsaicin, and for the TRPA1, we haven't tested any agonist in our chip. However, our supplier did test it previously.
Once again, that comes also on the characterization of the cells, and as we mentioned before, the cells being the heart of the model, really, in a certain way. We have to make sure that the cell we choose to use actually makes sense, and so we do rely a lot also on the quality control that is made by, and the calculation, I guess, that is made by the supplier.
Okay, excellent. Thanks both for your answers on that. We have, still about the organ-on-chip technology, generally speaking. It's basically how scalable is the organ-on-chip technology for high-throughput screening? So
that's actually a good question, I think. So it depends what we call actually high-throughput training. Screening. If you think about very, very early, discovery, when you still have like tens of thousands or like a lot of molecules to screen, well, that might not necessarily be the most adapted, of course. So it's maybe more a medium throughput type of thing. But the fact that this is actually one of the reasons why we decided to go with the SBS format, and so the 96 well plate format. The devices are compatible with any automatization device that you will actually have in your lab. It could be the automated cell culture robot or just a liquid handler, an automated liquid handler. It could also be, if you want to use imagery, for example, your automated imaging system, whether they be, I don't know, like Compartmentalization or whatnot. So the idea is the throughput is really coming from the fact that you can automatize the use of the devices, so you don't have to actually be there and do media changes and things like that by yourself. So you can actually have a lot of different devices running simultaneously.
Okay, excellent. We still have one question to go in the pipeline, so if you have any questions, feel free. How do you envisage the integration of this technology into current drug development pipelines?
Well,
who wants to enter that?
I can start, and maybe you can continue as well.
Okay.
I would just start because it actually linked to the question what I was saying before. I guess in terms of very, very, very early development might not be the best approach, but still, during the discovery part, when you still have a few tens, for example, of molecules, you can definitely start to use this kind of approaches to have an idea and make actually better choices. So have the good go and no goes. So either they be because you see effect, so efficacy or not, or that you can see because you know that this group of this family of molecules tend to induce various kind of side effects such as neuropathies, and you can actually start screening about which form or which formulation might be the one inducing less pain, to put it this way. And then it can actually get along your whole, it can go in parallel with your regular discovery and optimization pipeline. So even during lead optimization and even alongside actually your animal trials, in order to get more mechanistic information, more another kind of information, actually information that will be gathered on human cells. And potentially, so that's in the future, that's something we would like to do at some point, is to actually do that also in parallel of clinical trial in order to go a little bit more deeper, especially in order to get towards, I guess, better inclusion and exclusion criteria, so when you can actually start to screen beforehand the effect of a compound, or whether it be efficacy or even safety, once again, before putting it in humans. I guess there are things you can add on that. I don't know.
No, I think you did a really good job.
Okay, good. I'm getting better at this.
Yeah.
Thank you.
Okay. Thanks very much, both of you. I don't know if you have any some last questions from the audience. I mean, very lively, very interesting. So it's still possible to take a few questions. So apparently, my moderator tells me that there is no more question at this stage in the pipeline. We got some emails also already from this webinar, coming and asking us some extra data, so we will answer that on a one-by-one case, of course. So thanks very much for attending this webinar about MEA Pain on Chip platform. We really love the questions that you have asked, and we will, of course, give you access to this replay. Additionally to that, this is basically the summertime, so the webinar will start again in September at Netri, and we will basically inform you along the second half of August for the next webinars. And we have dramatic things coming over, and we hope that you will enjoy it. So thanks very much again, and have a great day.
Thank you.
Thank you.